About 1 in 6 individuals in the US binge drink. That translates to around 38 million individuals total. And of those individuals, about 10% have alcohol use disorder.
In Wisconsin, these rates are quite alarming. Recent numbers by CDC, or Wisconsin Department of Health Services, account for about 27% of Wisconsinites binge drinking, which is higher than the national average of 17%.
Wisconsin's trend is stubbornly high. We rank probably about top 10 in the nation for states that participate in binge drinking. And when it comes to trends, we see that for collegiate students, they actually tend to drink a lot more than what we expect. Surveys say that they typically participate in binge drinking at least twice a week or even more.
From an individual health standpoint, binge drinking results in a number of both acute and long-term related health problems. So on an acute time frame, we worry about things like accidents, injury, increases in violence, things like that.
Long-term alcohol-related health crises or deaths comprise of liver disease, cardiovascular disease, alcohol-related deaths, different types of cancers such as liver, colon, breast cancer, etc.
The CDC estimates that we spend close to $249 billion annually on costs related to alcohol misuse. And this includes things like lost productivity at work, things like hospitalization, emergency room-related costs, and motor vehicle-related deaths. So all of this really adds up to a very large societal cost as well as an individual toll.
Alcohol use disorder accounts to around 178,000 deaths per year, and that causes a significant economic burden on US as well as Wisconsin's economy.
Alcohol use disorder is, from a technical standpoint, something that's very treatable, and there are at least 3 different forms of FDA-approved medications that are available. However, there's a huge gap between the percentage of the population that has alcohol use disorder or that engages in binge drinking and that are actually getting some form of treatment.
There's pharmacological treatments that are FDA approved that are available for alcohol use disorder. Most people with alcohol use disorder do not have access to these types of treatments. And even when they have these sort of FDA approved therapies, relapse is still high and still a very prevalent issue.
There's also a lot of evidence to support that cognitive behavioral therapy is very beneficial for individuals struggling with alcohol misuse, or things like Alcoholics Anonymous, social groups where people can come in and have group therapy. So kind of the gold standard is a pairing of both medication and this behavioral therapy. But as I said before, really only 2% are getting both of those things for treatment. So a very large percentage of people that end up relapsing because of this. In fact, over 30% initially. And then as time passes, I think that number creeps more up to 70% relapsing.
In your brain, you have a very important region which is known as central amygdala, which plays a vital role in the vicious cycle of alcohol addiction. When a person goes through withdrawal, which is not having alcohol for a certain amount of time, this part of brain region becomes a hub for negative emotions such as stress, anxiety, and just wanting to seek alcohol. The negative effects and stress caused by alcohol consumptions makes this brain region sensitive to relapse, which is actually driving it back or motivating it for more alcohol consumption.
We're bringing a new perspective to the treatment of alcohol use disorder. Even though alcohol use disorder has been a problem for a very long time, we haven't had these new cutting-edge techniques available in our arsenal. So what we're doing is we're using these techniques such as single nucleus RNA sequencing, do exploratory analyses, and look at how changes in gene expression might be contributing to these underlying molecular changes in the brain.
So with single cell sequencing, we get individual cell level resolution, and we can pinpoint which genes are changing in which populations. And that will help us move towards more targeted cell type specific therapies. And I think ultimately that's going to lead towards more personalized treatments for alcohol misuse.
With this knowledge of imbalance, we could develop medications that target these pro or anti-stress modulators, which affects the cellular systems when you are most likely to be vulnerable for the relapse without affecting the healthy cells of your brain region.
What we're doing is we're asking the mice to drink alcohol and then looking at the behavior 72 to 48 hours after not having access and seeing from a molecular standpoint of view, what does stress look like on the brain when you have no access to alcohol.
From there, we take the information, the sequencing data that we get, we put it through computational pipelines that help us identify what cell types are present in the amygdala and what changes are happening in response to alcohol withdrawal in each of those populations.
Our goals for this project is to look at the central amygdala, which has a negative feedback loop when it comes to stress. So when you no longer have access to alcohol, that can put you in a stressful situation. And looking at that from a molecular standpoint of view, then we're hoping to see if we can use our CRISPR-based techniques, RNA-seq techniques. If we can get those to work, then we can implement that into drug use and then hopefully put those into clinical trials.
We define success as finding targets that we're going to try to target with CRISPR-based epigenetic editing tools, so to modulate acquisition of binge alcohol consumption behavior.
CRISPR is a revolutionary gene editing technology that recently received a Nobel Prize in 2020. If you want to know what CRISPR is, think of it as a scissor, but at a molecular level, where you can edit, cut, copy, and paste any gene target of interest. Scientists can use CRISPR to locate problematic genes and fix them or replace them with healthy versions of those gene targets.
Our team is uniquely qualified to tackle this issue because we specialize in single nucleus RNA sequencing and CRISPR-based approaches that allow us to tease apart what these changes in gene expression do on a cell type specific level and see how that affects behavior, specifically binge drinking behavior.
We also have established iPSC lines, which are basically stem cells that you would get from a healthy person or a patient to understand how rodent models translates into physiological or cellular changes in a human.
This funding, it's been beneficial in a number of different ways. So it's helping unite a number of investigators here at MCW that might have had prior experience doing alcohol work but weren't currently doing that work. And I think given the need and the binge alcohol problem that we have as a state, we really need to establish a foundation of investigators that are focusing on this. Hopefully in the long term, it's benefiting individuals who are suffering with alcohol use disorder if we can get a little bit closer to developing therapies and moving a little bit closer to cell type specific, more individualized, more personalized therapies to treat alcohol misuse. I think that's really important.